Yeah, well, I had the opportunity to go to Italy, and just about everybody was there. Um, it's a little bit harder to get to, so some of our researchers were not able to attend, but most of the labs sent at least one representative, so postdoc instead of the professor from the lab. And there were some significant advances in Fragile X research, and some of the things that really jumped out at me. I was the chair of the Therapeutics and Clinical Trials session in the meeting, and Dr. Steve Gray, who's at UT Southwestern, the same place as Kim Huber, who's the chair of the meeting, um, announced some really promising results in Fragile X gene therapy. So he's doing the traditional viral vector gene therapy in the Fragile X animal model, and he got some significant sustained expression of FMRP in the mouse model using the latest viral vectors that they've engineered. So that was really promising. I've talked before about the reactivation therapies that we've been working on, and I'll say a little bit more about that when I talk about our grants. But Joel Richter's work was presented by his star postdoc, who's now an assistant professor at UMass, Sneha Shah. And this is the first time that this has been presented at a general Fragile X meeting. And I would say that it was well received. It was met with a lot of enthusiasm, a little bit of appropriate skepticism. The point of having meetings like this is that you present your results and everybody gets to look at it and essentially try to shoot holes in your theory. And this is what you want. You want your theory to be well tested, especially if you're developing it into a therapeutic. And so that was presented and very well received. And it was also a great chance for us to meet a lot of our postdocs that we fund. A lot of the new grantees from FRAXA were there. We actually fund a fair amount of research in Italy, and a lot of the Italian scientists were there as well. So it's a great chance to see everybody and to hear the latest results. Unfortunately, you're not allowed to take pictures or send tweets from Gordon conferences. The motto of Gordon conferences is that, you know, what happens here stays here. we don't like you to send out dispatches from the meeting and tell everybody about the latest results while they're happening, or take pictures of the slides that they're showing. So we can't talk about a lot of specific results, but I can say that it's pretty exciting. And a lot of the folks in other fields, you'll notice the title is Autism-Related Disorders. A lot of the other folks in autism and rare diseases, rare neurodevelopmental disorders, really kind of envy what we're doing in the Fragile X field, because we're a lot further along than most of them. And so that's always a big topic of conversation, is how they can do things the way we've done things. And the research that we see coming from a lot of the other autism researchers is in fact often the kind of stuff that we did 15 years ago, and they're just getting around to doing it now. So we're in a pretty good position, and I think everybody has the feeling that a lot of these research topics are really maturing and they're really translating, and we're really on the cusp of some major advances in actual therapies. So I can talk a little bit more about that, but I think it's good to talk about some of the specific grants that we've awarded. As you probably know, we have a February 1st deadline for research grant applications every year, and so we typically make the awards during the course of April. And so we had a somewhat smaller batch of applications this year than we typically do, which means we gave out a somewhat smaller batch of grants. But we have a lot of other things going on, and so the basic science grants are just a part of what we do. But it's a way of loading the pipeline. It's a way of putting things in at the very beginning that will inspire innovation later on. So we'll start down the list. And of course, we've been funding Jeannie Lee and her efforts at reactivation of the Fragile X gene for quite some time now. We have just initiated another grant for Jeannie because she's making a tremendous amount of progress and is really beginning to commercialize this approach. There's now a company that's that's picked up the rights to this, and they're working on actually developing it as a treatment for Fragile X. Essentially, Jeannie started out as an expert in X inactivation. So that's the process whereby one of your X chromosomes gets inactivated. But if you understand how one of the X chromosomes gets inactivated, you have some insights already into how to reactivate that X chromosome. And so she's using that knowledge and that expertise to try to figure out how to activate the gene, the FMR1 gene, on the X chromosome in the case of Fragile X. Now, she's actually come up with two discrete methods for doing that along the way. And one of them is a small molecule method. It's basically using enzyme inhibitors that are used in generating induced pluripotent stem cells. And when you're generating stem cells from, say, blood cells, what you're trying to do is take a differentiated cell, so it's a cell that's already specialized, let's say a blood cell, and you're trying to take it into a cell that's no longer specialized, that is, a stem cell. So a stem cell is a cell that could become any other kind of cell. You can re-differentiate it into a different type of cell, like a neuron. So you can basically take blood cells and make them into neurons using this technology. Well, the way you actually do that is by inhibiting enzymes and essentially making that cell immature. And what we found out through experience is that immature cells during the course of development don't inactivate the Fragile X gene, even when there is this big CGG repeat expansion. And so she's found that if you use some of these enzyme inhibitors that are normally used to make stem cells, but you give them to Fragile X cells, you can actually start re-expression of the gene. In another line of research, she started looking at gene editing, and we all know about CRISPR. Now, CRISPR is often officially called CRISPR Cas9. Those are the two major enzymes that are used. They're bacterial enzymes. So you have the CRISPR enzyme itself, and then you have the Cas9, which is kind of a way of positioning the machinery to do the gene editing. So the Cas9 gets connected to a guide RNA, which is a section of RNA which mates up with the target section of DNA that you want to work on. So in this case, what Jeannie did is they started working up this procedure to use CRISPR, and they connected the guide RNA for the CGG repeats to the Cas9. And in this case, it's actually an inactive form of Cas9, because here they're just trying to position things right now. So they're trying to figure out how to position this and which guide RNAs to use. And what they found was that with the dead Cas9, if they put those into the Fragile X cells, that it actually contracted the repeat section. So the number of CGGs went from full mutation down into the pre-mutation range just by using this so-called dead Cas9 with an appropriate guide RNA. So now we have a very simple version of the CRISPR method. We don't have to get much stuff into cells, and that's really important in the brain. We don't have to get a lot of stuff in there to make this work. And we can also shorten the number of CGG repeats and get re-expression of the protein. So this is very exciting. The important thing now is to test how well this will work in non-dividing cells, like neurons. So all the cells in your brain, neurons and glia, are non-dividing. Unlike other cells in your body, they have to be able to do this process without mitosis, without cell division. And so that's what they're checking on now, is to see how well they can get it into cells and how well it'll work in non-dividing cells. And of course, the challenge after that is to try to test it in a whole brain. And the problem is that the mouse model doesn't actually, if you knock in CGG repeats into a mouse, it doesn't actually silence the gene the way human cells do. So you can't really model Fragile X very well in mice. But they're working on, there's a few little workarounds you can use for that, including grafting human cells into a mouse brain. And so they're doing some of those things right now as we speak, and we're funding that. We are also funding another lab at CHOP, Children's Hospital of Philadelphia, which is a very well-known outfit in the gene therapy field. Well, they're actually doing gene repair, gene editing using CRISPR and Cas9 in the Fragile X mouse. But in this case, they're using not the Fragile X knockout mouse because, well, that's just a knockout mouse. You've knocked out part of the gene. You really can't repair that. And that's not what we're trying to do in humans. But since we don't have that good mouse model for Fragile X, what they're doing as a stand-in right now is they're trying to correct one of the point mutation models of Fragile X. So some people, very rare cases, have these point mutations in the Fragile X gene. So one of the amino acids in the protein gets substituted and you end up having a non-functioning protein. So the reason for doing that is because you can actually test these methods, albeit with different sequences involved. Just think of those guide RNAs that I was talking about before, where you position this on the CGG repeats. Well, in this case, you would use different guide RNAs and you would edit the gene a little bit differently, but you're basically proving the concept in a mouse model where you can then show that you actually can do enough of this in a whole brain that you'll actually correct the behavior and the cognition of the mouse. And so this is an effort to perfect the techniques for actually getting this stuff into mouse brains. And once that technique is perfected and they've proven the concept, you can kind of just switch around the specific sequences of the CRISPR machinery that you're using, the guide RNAs, to do this a little bit differently. But once you've figured out the basic concept and tested the methods in the mouse model, we can presumably do this for any sequence we desire. And those are new folks. We always like new researchers in the Fragile X field, and those are people that are new to the Fragile X field. We're also doing a study with Karen Usdin at NIH, who has worked on gene reactivation in Fragile X for a very long time, but they've always gotten stuck. And of course, Joel Richter's research that FRAXA has funded at UMass has shown why that is. The reason why it's hard to reactivate the gene is, well, for one thing, the gene isn't really completely inactivated to start with, and it's producing this mutant splice variant that's actually interfering with synthesis of the protein. So what Karen Usdin is doing is a lot of the groundwork to figure out how it is that the FMR1 gene is silenced, or whatever it is you want to call it, why it is that it's not producing the protein. And she's going to be looking in human stem cells, where you actually can be looking at cells that have the CGG repeats. You're not looking at a knockout mouse model, which just has the gene the gene sort of cut out, or at least parts of it cut out. You're looking at human cells with the Fragile X mutation, and you're figuring out why this isn't working. And so basically, this is laying the groundwork for some of these reactivation studies and trying to figure out what the potential hurdles are that folks are going to have when they are able to repair the gene. How do we actually get it restarted and producing the protein? Another grant we're funding is a not entirely new researcher, but somebody we started funding a few years ago. A very highly regarded scientist named Lynn Marquardt is an expert on how RNA gets processed in cells, and specifically why it is that if you have a messenger RNA which has an error in it, your cells in your body have a mechanism, a quality control mechanism for weeding out those defective mRNAs, and it's called nonsense-mediated decay. And it turns out that in Fragile X, this is one of those processes that is excessive. So people with Fragile X do too much of this nonsense-mediated decay. We don't entirely understand why. It may very well be because, as Joel Richter has found, there is abnormal splicing in Fragile X. And so we're splicing things abnormally and inappropriately in a lot of cases, which generates lots of stuff that needs to be decayed or gotten rid of. But what Lynn Marquardt has found is if they inhibit this process of nonsense-mediated decay, then the animals actually improve. So they actually get better function. So it appears that a fair amount of this NMD is actually inappropriate, and it's too much. It's not simply a compensatory response to generation of abnormal mRNA. It's a process which all by itself has kind of run amok. And if you inhibit this, you can actually treat Fragile X. So we're continuing to fund that, and they're working on treatments based on that. We've also started a new grant to Mark Baer. We haven't funded Mark in a few years, and of course, everybody knows that Mark, along with Kim Huber, came up with the mGluR theory that led to large-scale clinical trials. A theory which is, by the way, as correct as it ever was. The mGluR theory is correct. It's just that the target is very difficult to treat. mGluR5 is just highly susceptible to tolerance, and that's all there is to it. So it doesn't make a good drug target, but the mGluR theory is correct. Well, now this is something that's a little bit different. They're actually going to be investigating interneurons, and these are the inhibitory neurons in the nervous system that control all of the excessive excitation in the brain. And as we know, in Fragile X, we seem to have too much excitatory neurotransmission and not enough inhibitory neurotransmission. And they have found a particular defect in a particular class of inhibitory interneurons, and that has specific ramifications for drug treatments that could enhance that particular class of interneurons. And so we have a new FRAXA fellow, Sarah Simpson, who I had the pleasure of meeting at the meeting in Italy. She's somebody who actually has a music background, and she's getting into neuroscience from a music background, which a couple of our researchers have. Len Kaczmarek at Yale is a world-class rock guitarist and an excellent musician. And so we have a number of people who've come at it that way. And Sarah is, as you might imagine, extraordinarily talented postdoc at MIT. And, you have to be pretty good to be at MIT. And she's very good. New researchers in Germany are going to be looking at the role of insulin signaling in actually regulating a number of other neurotransmitter systems, especially regulating dopamine. And this is really exciting because this is the crossover between metabolism and neurotransmission. We all know that what's going on in your body affects what's going on in your brain. And one of the primary ways it does that is through insulin signaling. This is something that hasn't really been studied quite enough, I think, but a lot of the research in insulin signaling has been very productive so far. And in fact, that's why we have these large-scale metformin trials right now, and why a lot of people are taking metformin for Fragile X is because of the understanding that insulin signaling is dysregulated in Fragile X. And so these folks are looking at one of the facets of dysregulated insulin signaling in controlling dopamine expression in the parts of the brain that regulate a lot of the cognitive rigidity or cognitive flexibility, which is one of the hallmarks of Fragile X and also one of the hallmarks of autism. Jinyu Zhao is another superb scientist at the University of Wisconsin that we funded a fair amount in the past, and we have a new grant going to her lab that's going to actually investigate the developmental timing of treatments. One of the biggest questions we get from pharma companies, and one of the biggest questions we get from parents that we've already gotten, is can you use this or that treatment at this particular age, or do you have to do it really early on? You can do studies in the animal models to try to figure that out, and that's exactly what she's doing. And this is designed to answer a lot of those questions that gene therapy companies have. If you want to know, well, can we do gene therapy in 30-year-olds with Fragile X, or is that just a waste of time and we need to do it in 2-year-olds? She's going to try to answer some of those questions, but she's also using a particular focus on mitochondrial function. And again, this is that metabolic aspect of Fragile X that has been ignored for a long time and now is a fairly hot topic. A lot of people are realizing that it's the energy production in the brain. Your brain it's the most energy intensive organ in your body. About a third of all the energy that your body uses goes to the brain. And the energy is produced in these little organelles, these little teeny parts of the cell called mitochondria. And evolutionarily, mitochondria actually derived from bacteria. So it's sort of like you had bacteria invading your cells and set up shop, and now we all live together and we require those bacteria to make our cells run. Well, in Fragile X, you've got so much requirement of energy from cells in your brain that you're really stressing out your mitochondria. And you can actually observe mitochondrial dysfunction and abnormalities in the mitochondria if you look closely. Now, we've had several labs recently that have worked on this, and everybody's coming at it from a little bit different angle, but they're all finding results that line up pretty well. And that is that if you can rescue these problems with the mitochondria through various strategies, then you hopefully drug strategies or other kinds of treatments, potentially diet and lifestyle strategies even, then you can actually rescue the overall behavior and cognition of the animals. We've got a clinical trial with Dragana Proteome, who is an MD, PhD behavioral pediatrician, developmental pediatrician, I should say, in Serbia. So she's at the University of Belgrade, and she's studying probiotics and the microbiome, in other words, the bacteria in your gut, and looking at how that may help with behavior in kids with Fragile X. This is a small study. This is designed to be a pilot trial that will pave the way perhaps for something larger, or if the results are good enough, then hopefully will actually change clinical practice somewhat. This is a hot topic as well, the microbiome in Fragile X, and it is relatively easy to observe differences in the gut bacteria of kids with Fragile X. The question is whether that's cause or effect. Is it because they have kind of idiosyncratic diets, let's say, or is it because there's something about Fragile X that changes the environment for the bacteria? And so we're trying to do those studies, but one way to tease out the difference between cause and effect is to actually do some kind of intervention and to try to change the gut microbiome and see if that helps with behavior. It's really important, I think, to develop new trial sites, especially outside of the US, because we've got clinical trials going on which have a very good chance of succeeding. And if they succeed, we have a lot of pharma partners that we're working with who are about to jump into the Fragile X field with the drugs that they're developing. And the problem is we only have certain clinical trials capacity, and we only have certain number of trial sites, and we need to develop more of those trial sites. And so we're trying to do that. And the best way to develop new trial sites is to fund clinical trials. Last but not least, along the same lines of trying to enhance our clinical trials capacity, one way to do that is to allow people to do clinical trials at home. The current clinical trials involve things like the NIH toolbox, electrophysiology, eye tracking, all kinds of stuff that requires a pretty sophisticated setup. But that's not necessary, and it is possible to use simpler technology to accomplish the same thing. And so we're funding Craig Erickson and his postdoc, Lisa DiStefano, to study EEG at home in Fragile X subjects to see if we can get results that are as good as doing the EEG in the clinic. And that's really important because if we can fill in all those gaps on the map where people are with Fragile X, but there aren't any trial sites, then we can start recruiting lots more people, even right here in the US. We don't necessarily have to go to China or India to get more trial subjects. We can actually find them right here at home, but they're folks who might not be able to make it into the clinical trial sites. And I'm sure a lot of you have kids who don't travel very well. And I think that's a real problem in clinical trials. For pretty much any disorder, you don't always get a representative sample of the population. And by going out and treating people or doing the clinical trials in home, you end up getting a much more representative sample. It's even possible that you get a better result because we often talk about the high placebo response rate in Fragile X trials. But I really don't think it's just placebo response in the conventional sense where we think of people feeling better because they're taking a pill, so it's kind of all in their head. No, that's not really what's going on. I think what's going on in Fragile X clinical trials is our kids will go into this trial site. Often it's a place they've never been before, and they're very uncomfortable there. But as they go over and over again, a few times, in the case of our son, 3 or 4 times, and he's starting to like it. He's not nervous about going there. He's looking forward to going there. And so they have this habituation to the stressful environment of the clinical trials site, which comes across as a placebo response when they start to look at the rating scales. So if you can minimize that by actually going out and rating people in home, you can hopefully eliminate that component of the placebo response. So I think this is a very important technology to develop, the idea that we can go out and evaluate people in their homes. And if we can do this kind of electrophysiology in their homes, that's even better.
Excellent. So just a reminder, if you guys have any questions, you can use the Q&A feature at the bottom. We did get a few submitted in, so I'll run through them. First up is, I was curious what the status of research into ASOs.
Yeah, as I had alluded to before, and we talked at length last time about Joel Richter's groundbreaking study, and this is really in the finest tradition of medicine, a completely serendipitous discovery. He started out by observing that there were abnormal splicing events going on in Fragile X cells. In other words, when the messenger RNA is made from the DNA, from the chromosome, when it's transcribed, it is then subject to splicing. And I make the analogy to the movies. You know, the director may have his director's cut of the film. They splice things together to get different versions of the film, and they often have they splice things together differently for different countries or even different parts of the country. Well, it's the same thing with a messenger RNA. You may have one gene, but you can splice that gene up a number of different ways. The splicing doesn't happen with the DNA, it happens with the RNA. So the RNA gets spliced, the copy gets spliced, and then it gets exported to the cell, and it gets spliced differently in different cells. And that's what makes different cells different, is they get differently spliced genes, differently spliced mRNAs. So Joel observed this difference, and FRAXA funded a study to look at this as a potential biomarker. And one of the hallmarks of this study, I think, was that it was the first one to ever really look in depth at with RNA sequencing. And we're talking about approximately $50,000 worth of RNA sequencing. So very expensive stuff had to be done to assess these aberrant splicing events. But along the way, what he found was one of the things that was misspliced was FMR1. But the thing is, he's looking in Fragile X cells, and he's finding misspliced FMR1, when there shouldn't be any FMR1 at all. So he's wondering, well, how is this? Then he looked further and he found that in a lot of these cells that had misspliced FMR1, so the gene was not silenced, but is producing this abnormal version of the messenger RNA. It's also producing lots of the normal version of the FMR1 mRNA. So you're producing the messenger RNA for the Fragile X protein, but these same cells had no protein. So how is it that you're producing lots of messenger RNA, but you're not producing any protein? And by the way, you're also producing this abnormal splice variant of FMR1. Well, he wasn't sure exactly why it was, but the first thing that occurred to him was maybe this abnormal splice variant is blocking the production of the protein with this normal mRNA. So they made ASOs, these antisense oligonucleotides. Again, these are sections of genetic material that pairs up perfectly with the sequence that you're trying to target. So he made a bunch of different ASOs, different parts of this abnormally spliced FMR1 mRNA. And what do you know? He saw that when you introduce these ASOs, you actually got expression of the protein. So these cells that weren't producing any protein are suddenly producing normal levels of protein. And it's about 3/4 of the blood cells, the blood samples that they've looked at so far that produce this abnormal splice variant, so that have the target. And you might wonder, well, what if you're one of the quarter of the population that's not producing that? Is there any hope for you? Well, it turns out that if they simply gave a simple demethylating agent to those cells, those blood cells, they would start producing the abnormal splice variant, and then you could put the ASOs in and they would start producing normal amounts of protein. So you have kind of a two-step process for a quarter of the population, but a simple one-step ASO for 3/4 of the population. And ASOs are a little bit different from gene therapy. They're very long acting, but they're not permanent. And it's not a one-shot deal. You have to administer them about every 2 to 3 months. Sometimes more, sometimes less. And it depends. Sometimes you start out more and then you spread them out as you go. And so there's actually a company that has picked up on this and is working with the Richter Lab and UMass. Details are all confidential at this point, so I can't really say much more than that. But I can say that there was a representative from that company at the GRC in Italy, so they are serious about this.
Excellent. All right, this question was for Katie, was what is the best way to learn and track what they are doing?
If you want to get the real scientific details and don't mind reading those papers, you run on over to pubmed.gov and then plug in any researcher's name, comma, Fragile X, or try your own searches the way you want them, and you'll probably come up with some papers that they've written. Unfortunately, there's a lag time between discovery and paper, and they are some heavy duty reading, that could put you to sleep at night. You can also subscribe to FRAXA's emails or go and plug in that same search in fraxa.org, because we have, 400 pages, 400 teams up there. Each one has the details of their projects. We are in the process of really spiffing that up, which is a lot of work, as you might imagine, because this is 30 years of, publications on the website. But they are there. So take a look, if you want to find out about ASOs, go to fraxa.org and type in ASO in the search bar and have fun. So we try to write them for, the rest of the world, but PubMed has the definitive papers.
Plus, we got there's a really nice independent site called the Fragile X News Today. And you can sign up for that and subscribe to them. And they often pick up a lot of the things that we do, and they often pick up on a lot of the publications from our researchers as well. But they have a really good team of science writers who make this understandable. So they do a really nice job of it. And for any of you who haven't discovered Google Alerts, just put in a Google alert for Fragile X. You'll get stuff in your email every day.
Question related to CRISPR Cas9. What's the status of current work on it, and when could we expect the first phase of a clinical trial?
Well, that's a good question. I'd say Jeannie Lee's strategy to use the dead Cas9 is the simplest and most elegant, and a lot of the sort of go, no-go kinds of basic studies are being done right now to see if that's going to actually work as a therapeutic strategy in humans with Fragile X. So it's a little too soon to say when that might actually get to the market. I think it's considerably more likely we'll see the ASO treatments before that. I think these antisense oligonucleotides they're in trials for a lot of different conditions. The technology is getting pretty well developed. It's not something that's easy to administer. You basically have to do it it's kind of like having spinal anesthesia or a spinal tap every time you get it. But Liz Berry-Kravis in Chicago is working on 2 Angelman syndrome ASO trials right now. And she's the one who actually administers the stuff. So she actually gives the doses. She's a neurologist after all, so she's very good at doing spinal taps. And so she says it's not bad, and people stay with it, and they're in the trials, and they don't mind doing it, and they get used to it. It's like anything else. It's hard at first, but they don't mind it, and they keep coming back. So that's good news. It sounds awful, but it's not as bad as it sounds.
Because we've got a few more questions, and we'll actually open it up. So if you guys have questions about anything else related to the Fragile research, you can feel free to drop those. Again, use the Q&A button at the bottom. We did have a good one come in. Do we have a credible combination of drugs that will reduce the symptoms of Fragile X in the next few years?
Well, the answer is yes, we've got lots of combinations. The problem is it's really hard to do clinical trials of combinations. So combinations are something that doctors come up with over time based on clinical experience. When a drug company actually wants to get a combination of drugs approved, they have to do a very complicated, large, and expensive kind of clinical trial to get that approval. Right now, as you probably know, we've been working with a repurposing company called Helix, which is based in Cambridge, England. And they've got a combination of drugs that are, technically speaking, well, one of them is an available drug called Ibutylast. Problem is it's available in Japan, not the US. The other is a drug called Gaboxidol, which is the same drug that was in trials with Ovid. And so some of you may have even been in the Gaboxidol trial for Fragile X. It showed some promising results, but some mixed results. The combination of Ibutylast and Gaboxidol in the Fragile X mice works really well. And the scientists involved in that published a paper recently describing that. I was actually a co-author on that because I was involved in the preclinical testing. So the drugs complement one another very well. So they make an ideal combination because one does a certain set of things and the other does a certain set of things. They offset each other's side effects to some extent. They're just a really good combination. In the animal model, it worked great. There's no reason why you couldn't use it in humans, but the FDA has a little bit of a problem with just going ahead and doing a clinical trial of 2 technically investigational drugs because the FDA has not approved either of these drugs for general marketing. And so they're actually going to be going and the company may well do a pilot study in another country where maybe the regulators there don't have as much of a problem with doing the 2 investigational drugs, or where Ibutylast is actually approved. So it would only be 1 investigational drug in combination with an approved drug, which is much easier. So that's kind of a long-winded way of saying that, yeah, we've got a lot of combinations, but it's really tricky to actually prove which ones work the best. It's easy enough for me to just prescribe a combination of meds and say, oh, this works great, I'm going to give this to everybody. But to prove that and to convince other people to prescribe that is another story entirely. And then to get something like that approved for marketing as a kind of brand name drug is more difficult still. There is a company called Stalicla, which is based in Switzerland, which is also looking at Abutylast in combination with bumetanide for autism. And they actually did a small pilot study with Craig Erickson in Cincinnati, and they were looking for people who could do the kind of clinical trials that we've been doing. And so I actually introduced them to Craig Erickson, and he's very excited about the results, and they should be published soon. But the company has a certain phenotype that they look for. So it's kind of like a 10-point checklist of symptoms and lab tests that will indicate that this particular person with autism is more likely to respond to this drug combination. So it's a really nice strategy. And most people with Fragile X would qualify according to that, what they call phenotype 1. They have a number of different phenotypes and a number of different combinations for those phenotypes. And so this is really the way forward, at least according to some folks in the autism and Fragile X field, is to have this kind of phenotyping or some way of telling which profile you have, and that will indicate which combination of drugs you're better off taking. But unfortunately, the trials are hard to do, and the FDA has a lot of questions about this.
I wanted to pipe in there. When you've got a combination of drugs, you also have to figure out what dose of each drug you're going to use and whether those two medicines influence each other. You know, if they're a combination, maybe they are for a particular reason. And this whole issue of dosage has been probably confounding results of the or people's experiences with the current study meds. You know, for example, the drug from Tetra, you know, for our son, we felt like he was getting benefit, but then he had a side effect. Well, maybe what he needs is a much smaller dose. We don't really know. It's so early, you can't tell. Somebody else who's having no side effects, but not much effect, you know, good effect either, maybe they need a larger dose. So it's just, you know, it's gets complicated when you add more drugs into a combination.
Yeah, one size doesn't fit all. That's the problem. And the reason why drug companies are often stuck with this methodology of prescribing, of giving everybody in a trial the exact same dose, is because if they wanted to do 2 doses, then they basically have to have twice as many subjects in the trial. And if it's hard enough to do the trial the way it is, and if it's going to take Tetra a couple of years to do these trials, you can imagine if they needed to do twice as many subjects to have 2 dose groups, well, then it would take forever and you'd never get anything approved. But when you have combinations of drugs, then you have the sort of the ratio of the 2 drugs, which is then fixed by the combination if you have an actual combination pill. And I'm not sure if the person asking the question was really thinking about it in these terms, but because, you know, we do have nice combinations of drugs that we can use in clinical practice. And in psychiatry, everybody's on multiple drugs. It's not at all unusual for people to be on. If you've got bipolar disorder, you know, you're on average in the US, you're on 4 or 5 different drugs. And so we always use combinations for these major forms of psychiatric disorders. And Fragile X would be no exception to that. But there are actually combination pills, and pharma companies were really enthusiastic about producing combo pills back in the day. And so in the 50s and 60s, they put out all of these combination pills like Triavil, that was a combination of an antipsychotic and an antidepressant. And there were a whole bunch of different variations on that theme. And I think the FDA got progressively more skeptical about that whole idea. And there hasn't been a combination pill approved in psychiatry in the last 20 years or so.
Excellent. All right. You mentioned Helix not too long ago. There was actually a question that came in just before you said that. And they were asking if AI can help in finding treatments for Fragile X.
Yeah, there are a number of companies that are using AI in a number of different ways. The most obvious way is just to look for repurposed drugs, for drugs that might have other effects besides the one that's the primary indication that might make them useful for Fragile X. But AI can be used in drug design as well. And so we're actually working with a company in Belgium that uses AI to predict structures of drugs, and then they will generate these drugs. So it's a completely new drug. They design a novel drug based on the structure that they think they need. But they can also then give that to animals and look at the responses, and then they can actually, using an iterative process, tweak that molecule and try to optimize it to get the right kinds of behavioral effects from the molecule. So there's a lot of different ways that you can use AI. You can use it for designing new drugs. You can use it for repurposing old drugs. You can also use it for this phenotyping that I was referring to that Stalikla is doing. You know, we have a lot of knowledge. We have a large set of data in Fragile X. There's a lot of research that's been done, and it's already to the point where no one human being can really synthesize it all and make sense of it. But that's the nice thing about AI and machine learning, is that you can kind of design an algorithm that'll chew through all the world's literature and get everything in there and try to make use of all that data that's already been generated. So that's another way to do it. And the Stalikla is using that to design their phenotypes of different subjects. And they're actually using trial results. So for example, in the R-baclofen trials, it was just to grossly oversimplify it, I would say that we saw about a third of the people had great responses to R-baclofen, about a third of the people had no response to R-baclofen, and about a third of the people had really disastrous bad responses to R-baclofen. So the question is, what separates those three groups of people? What's distinctive about the responders versus the non-responders? And so the AI can actually make sense of that. And I think personally, I think that's probably the greatest value of AI, is to try to tease out some of these variables and help us to stratify the patient population so we can understand, okay, now we're going to do another trial of R-baclofen, but this time we think we know what distinguishes the responders, and we're going to make that an inclusion criterion. And it might be electrophysiology. You might have to get an EEG ahead of time. It might be something like the NIH toolbox, where you do some performance task on a tablet. That's what distinguishes you. It might be a series of questions that you ask to the caregivers. It might be something about your medical history. It might be a blood test. We don't really know going in until the AI spits out that kind of result. And of course, we have to test that over and over again. But when we're dealing with people, we're dealing with enormous complexity. The researchers at meetings like the GRC that we just had are constantly talking about the differences between different strains of mice whenever we're doing these animal studies, and how it all depends on which, whether you're using C57s or FVBs and all these different strains of mice. You get different results. They could all be Fragile X knockout mice, but you get very different results with different strains. Well, just imagine how many different strains of human beings there are. We are a very, very mixed background, that's for sure. And so it's really important to be able to get a grip on that variability in the human population. And I think that's really where AI is probably going to have its greatest impact.
All right. Here's a fun what-if question. If Fragile X is the cause of autism in a person and Fragile X is cured, is autism then cured?
Absolutely. Yes. Autism is a symptom. Autism is not a disease. Fragile X is a disease, and Fragile X is a disease which often causes autism. Just as Fragile X, the disease, causes seizures in some people. If you have a disease-modifying treatment, you would expect that the seizures would at least improve, if not completely go away. And you would expect any autistic symptoms to improve, if not completely go away. Definitely.
Here's another. We've got, for family members of midlife adults with Fragile X, what would you consider reasonable expectations for improvements, and how much could a treatment or a cure help them?
You know, it's always hard to know exactly how much anything's going to help older folks compared to younger folks. And we kind of touched on this subject a little bit before. But I think it's very reassuring to understand that most, if not all, treatments are going to be studied first, and they're going to be studied the most in adults. Anything that gets to the market is going to have to work in adults because it's going to be tested there first. And if it's tested in adults and it doesn't work, they're not even going to go on to the pediatric testing. I mean, that's just the way the pharma protocols go. You know, to their way of thinking, we have to start with adults because that's what's ethical. We don't want to mess around with little kids. There have been other diseases. SMA is a perfect example, spinal muscular atrophy, where in severe forms, kids get very sick when they're very, very young, and they often will die before their 10th birthday. And so they have to start treatment really early because they'll be dead. And once you're dead, and there's no bringing you back. But people with Fragile X aren't dead. And people worry a lot about critical periods of development. First and foremost, I think it's important to remember that this concept of critical periods in brain development, this notion that, you know, you develop your visual cortex between the ages of such and such and such and such, that's a much more prominent effect in animals than it is in humans. Humans have a much more plastic brain. Second of all, we're born much less mature. I mean, your nervous system doesn't fully mature until you're in your early 20s. And I'm sure anybody with teenagers can attest to that. I mean, we all know that's true, and it is actually scientifically true. It just doesn't just look that way, it is that way. The nervous system is continually developing in human beings. But okay, let's say, what about after 30? Well, the brain retains a huge amount of plasticity. For example, if you have a stroke when you're 30 or 40 for that matter, a lot of the functions of that area of the brain, this is essentially like a brain attack, that like a heart attack kills off a little chunk of heart muscle. Well, a stroke kills off a section of the brain, and that whole section of the brain that's usually supplied by one blood vessel just dies. And so nothing in that part of the brain works anymore. But people who have a stroke when they're in the 30s or 40s can often shift all of the functions of that part of their brain to another part of their brain. So there's functional plasticity in the human brain that's enormous. Now, in Fragile X, obviously, something's not working right, and it's not working right throughout the whole brain. But if you can actually restore that proper function and at least get it within the normal operating range, then there's no reason to think that those functions can't actually be restored to most parts of the brain pretty well. I think what we'll probably end up seeing, assuming we have our gene therapy or we have our ASOs or whatever definitive therapy comes along first, what we'll see is that it'll be tested in adults, and it'll clearly work, and people will gain cognitive function. Their mood will improve. All the behavioral symptoms will improve. They'll become much more cognitively flexible. And whereas cognitive rigidity is kind of like the hallmark of Fragile X and autism. So you'll see that improvement in their ability to just deal with regular everyday life. But it'll come along gradually. It'll come along fairly slowly. And that's kind of what we're seeing in the Tetra trial in the adult population, is that people are reporting good results, but it's not like it starts right from the first pill. It takes people a while to catch up, and it just gets better and better as you stick with it. But I think what we'll see is that if we have treatments like this that start in adults, when we start doing the pediatric trials, the kids will have a much more rapid response to it. they'll just respond much more quickly, much more completely. It just won't take a really long time for them to do that kind of rewiring and rehabilitation that is required in adults. And I just think of it in terms of the kind of the stroke model. You know, there's a period of rewiring and rehabilitation that's required, and our kids are certainly going to need that after they've been given a definitive treatment of some sort, whether it's gene therapy, ASOs, protein replacement, whatever.
All right, here's another one. We've got, which research group is getting closer to having a cure out there for the public?
For Fragile X, I assume they mean, yeah. And I definitely, I think we've got a couple of groups that have made major progress in gene reactivation between Jeannie Lee and Joel Richter. They're doing fantastic work. I think Joel's is a little bit further along and is, you know, the ASOs are a technology that has been reasonably well developed at this point. I mean, it would be ideal if you could just take it in pill form, but you know, as it is, it's pretty good. And it really is just a matter of swapping sequences to get it to work. I mean, the basic idea is pretty much the same no matter what the disease. You just change the actual sequence of the oligonucleotide. So that is pretty much ready to go if they can start to get some proof of principle in non-dividing cells, which is what they're working on now, human neurons, and they're using these organoids. And of course, we have this technology to then take those organoids and transplant them into a mouse brain so that they can grow and develop. And so then you can try treating the whole animal by doing the infusions into the cerebrospinal fluid. And you could just test every single stage of before you actually take it into humans. But this is the type of treatment that's being used in humans. And another thing, as I mentioned before, ASOs are not permanent. They are reversible. So if you really mess something up, if something goes horribly wrong, it's not permanent. Whereas like a viral vector gene therapy, it's more or less permanent. So you have to be sure it's going to work before you put it in there. And if you do put it in there and something goes wrong, it's really, really bad. So if you have one adverse event in the first couple of patients, the whole program gets shut down. Whereas with ASOs, there's a lot of tweaking that you can do, and you can dose it individually. You can increase the dose, decrease the dose. It's really pretty simple, and it's something that's being done in a lot of human clinical trials right now. So that's something as far as what I like to refer to as the definitive therapeutics, the things that actually treat Fragile X directly. ASOs will almost certainly be first at this point. It's also possible that we might have protein replacement treatment as well, because Ray Turner, who's one of our other grantees in Calgary, in Canada, has had a really kind of interesting insight in the protein replacement. You know, if you just stick FMRP into a bunch of cells willy-nilly, it's really not good for it. It's kind of toxic because it messes around with protein synthesis and vital functions of the cell. But Ray's insight was that we don't have to put the whole protein in, that we can just put part of the protein in, and it just does some of the things that FMRP does. It doesn't do everything. It doesn't completely fix the whole problem, but it fixes about half of it. And you can put this part of the protein in without messing up protein synthesis and without causing any kind of toxicity. And at least in the mice, he can put it in the tail vein and it gets into the brain and it has actual behavioral effects, and it lasts about 2 weeks after a single administration, which is pretty cool. So it is conceivable that we could have a protein replacement therapy fairly soon. And I like to see competing strategies. And so now we've got several competing strategies. You know, I see our job at FRAXA is to make a horse race out of this. We want to have competitors. We want to have people racing to the finish line because that's where we get the payoff. And so we've got a little bit of a horse race when it comes to these definitive strategies now. We've got competing strategies for reactivation. We've got protein synthesis. We've got 6 or 7 companies that are interested in gene therapy for Fragile X. And we're also doing the gene editing that we've talked about. So it's pretty exciting.
And that's a great note for wrapping up. We didn't answer how long will it take, because as Mike likes to say, it's really hard to predict the future when it hasn't happened yet. But may the
Well, hard
best
to predict stuff,
win.
especially in the future. Yeah.
And yeah, and just think about, injecting into the tail vein. We should be all set once we grow tails, right?
Or an IV anyway. At least that's a little easier to take.
That's a lot better than the spinal tap.
Yeah.
Well, thank you, everybody. Really appreciate you taking the time and sticking with us and all this time. We're pretty excited about what's going on and hope to see something really good coming not too distant future.
All right, thanks everyone. Bye.
Bye.
Bye.